Biology Exam Preparation for Medicine Students Part 1. Which of the following is not a component of the endomembrane system in eukaryotic cells?
The correct answer is peroxisome. The endomembrane system in eukaryotic cells includes organelles that work together to modify, package, and transport lipids and proteins. Key components are the endoplasmic reticulum, ER, the Golgi apparatus, lysosomes, vesicles, and the nuclear envelope. The peroxisome, however, is not part of this system. While it is an essential organelle involved in breaking down fatty acids and detoxifying harmful substances like hydrogen peroxide, it operates independently of the endomembrane system. Peroxisomes do not receive vesicles from the Golgi apparatus, unlike lysosomes. To contrast, the lysosome is a component of the endomembrane system and serves as the cell's recycling center, breaking down waste materials. The Golgi apparatus processes and packages proteins and lipids, while the ER synthesizes and modifies these molecules. Understanding this distinction highlights how different organelles collaborate to maintain cellular function, each fulfilling specific roles. The independence of peroxisomes makes them unique among organelles.
Which enzyme is responsible for relieving supercoiling ahead of the replication fork during DNA replication?
The correct answer is topoisomerase. During DNA replication, the unwinding of the double helix by helicase creates tension ahead of the replication fork, resulting in supercoiling of the DNA. If not relieved, this supercoiling can hinder the progress of the replication machinery. Topoisomerase plays a critical role by cutting one or both strands of the DNA, allowing it to unwind and release the tension. Afterward, the enzyme reseals the DNA strands, ensuring the structure remains intact. This action makes topoisomerase essential for maintaining the integrity and efficiency of DNA replication. In contrast, helicase is responsible for unwinding the double helix by breaking hydrogen bonds between complementary bases. DNA polymerase synthesizes the new DNA strand by adding nucleotides to the growing chain, and primase synthesizes RNA primers that DNA polymerase requires to initiate replication. Each enzyme has a specific function in the replication process, but without topoisomerase, the replication machinery would stall due to the physical strain on the DNA.
In photosynthesis, which molecule is the first stable product of carbon fixation in the Calvin cycle?
The correct answer is 3-phosphoglycerate. During the Calvin cycle, carbon dioxide is fixed by the enzyme ribulose 1,5-bisphosphate carboxylase/oxygenase, commonly known as rubis-CO. Rubis-CO catalyzes the attachment of CO2 to ribulose 1,5-bisphosphate RuBP, forming an unstable 6-carbon intermediate that quickly splits into two molecules of 3-phosphoglycerate, 3-PGA. In contrast, glyceraldehyde, 3-phosphate, G3P, is a later product of the Calvin cycle, formed after 3-PGA is phosphorylated and reduced. Ribulose, 1,5-bisphosphate, is the molecule that reacts with CO2, not the product, and glucose is synthesized only after multiple turns of the Calvin cycle. Understanding this pathway emphasizes the central role of 3-PGA as the first stable intermediate in carbon fixation.
Which of the following best describes the role of cytochrome C in cellular respiration?
The correct answer is transfers electrons between complex 3 and 4. Cytochrome C is a small heme protein located in the intermembrane space of mitochondria. It plays a key role in the electron transport chain, transferring electrons from complex 3, cytochrome BC1 complex, to complex 4, cytochrome C oxidase. This electron transfer is essential for the chain to continue and for the generation of the proton gradient required for ATP synthesis. It does not directly facilitate proton pumping in complex 3, as that is a function of the complex itself. Nor does it transport electrons within complex 4 or synthesize ADP. Its role as an electron carrier is critical for oxidative phosphorylation and energy production.
Which RNA molecule is responsible for splicing out introns in pre-mRNA?
The correct answer is snRNA. Small nuclear RNA, snRNA, in combination with proteins, form small nuclear ribonucleoproteins, snRNPs, which are the functional units of the spliceosome. The spliceosome is responsible for recognizing and excising introns from pre-mRNA, thereby producing mature mRNA that can be translated into proteins. In contrast, tRNA is involved in translating mRNA into protein. miRNA regulates gene expression by silencing specific mRNAs, and rRNA forms the structural and catalytic core of ribosomes. Splicing is a critical step in eukaryotic gene expression, and snRNA plays a central role in ensuring the accuracy and efficiency of this process.
During meiosis, what is the term for the physical exchange of genetic material between homologous chromosomes?
The correct answer is crossing over. Crossing over occurs during prophase I of meiosis, when homologous chromosomes pair up and exchange segments of genetic material. This process creates new combinations of alleles on chromosomes, contributing to genetic diversity in offspring. Independent assortment, on the other hand, refers to the random orientation of homologous chromosome pairs during metaphase I. Synapsis is the process of homologous chromosomes pairing, while nondisjunction is an error in meiosis where chromosomes fail to separate properly. Crossing over is key to creating genetic variability, a vital aspect of sexual reproduction.
Which of the following processes requires the import of pyruvate into the mitochondria?
The correct answer is pyruvate oxidation. After glycolysis in the cytoplasm, pyruvate is transported into the mitochondrial matrix, where it undergoes oxidative decarboxylation. This process, catalyzed by the pyruvate dehydrogenase complex, converts pyruvate into acetyl-CoA, producing NADH and releasing CO2 as byproducts. Glycolysis occurs entirely in the cytoplasm and does not require mitochondrial transport. Fermentation happens in the absence of oxygen and does not involve mitochondria. The Calvin cycle is part of photosynthesis in plants and occurs in the chloroplast, not the mitochondria. Pyruvate oxidation is a critical link between glycolysis and the citric acid cycle in cellular respiration.
What is the primary function of ribosomes in the cell?
The correct answer is translation. Ribosomes are molecular machines responsible for synthesizing proteins by translating the genetic code carried by mRNA into a specific sequence of amino acids. This process involves binding mRNA and recruiting tRNA molecules carrying amino acids, which are joined together in the sequence dictated by the mRNA codons. Transcription, on the other hand, is the synthesis of RNA from a DNA template and occurs in the nucleus. DNA replication copies the genetic material, while splicing removes introns from pre-mRNA. Ribosomes are essential for producing the proteins required for all cellular functions.
In plants, which hormone is responsible for promoting seed dormancy and drought resistance?
The correct answer is abscisic acid. Abscisic acid, ABA, is a plant hormone that plays a key role in promoting seed dormancy, preventing germination under unfavorable conditions. It also helps plants respond to drought stress by inducing stomatal closure, reducing water loss through transpiration. Auxin primarily regulates cell elongation and growth. Cytokinins promote cell division, and ethylene is associated with fruit ripening and stress responses. ABA's role in stress adaptation and seed dormancy makes it crucial for plant survival in challenging environments.
Which structural feature allows tRNA to bind to its corresponding amino acid?
The correct answer is acceptor stem. The acceptor stem is the 3-foot end of the tRNA molecule where a specific amino acid is covalently attached by an aminoacyl tRNA synthetase enzyme. This attachment ensures that the tRNA can deliver the correct amino acid during translation. The anticodon loop interacts with the mRNA codon, ensuring accurate pairing during protein synthesis. The D-arm and variable loop contribute to the structural stability and recognition of the tRNA molecule, but they do not directly bind the amino acid. The acceptor stem is critical for the proper functioning of tRNA in translation.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.